In this section, we will cover the remaining part of Chapter 12. Your book talks about action potential, and I have left a video for you guys to review. It's approximately 14 minutes just to go over the action potential that occurs within a neuron. So please take some time out and review that video. So we'll move on talking about the synapses. Now, a synapse exists at the end of an axon. In synapses between two neurons, the first neuron is the presynaptic neuron, and the second is the postsynaptic neuron. With the presynaptic neuron, it releases neurotransmitters, and the second is the postsynaptic, and that is the one that responds to the neurotransmitter. Now these synapses, they trigger the release of neurotransmitters, and they also stimulate a new wave of electrical activity in the next cell across the synapse. So here, we're going to look at the diagram. Here is your presynaptic neuron. And here is your postsynaptic neuron. Here you have various vesicles that will carry the neurotransmitters, and your various channels on the presynaptic neuron. On the postsynaptic, you're going to have receptors that are going to receive the neurotransmitter that's being released, and then it'll travel to the up the postsynaptic neuron. Now, a presynaptic neuron may form an axodendritic, an axosomatic, or an axo-axonic synapses. So the axodendritic is a synapse between the dendrites. The axosomatic is a synapse between the soma, and the axo-axonic is the synapse between the axon, hence the names. Now, a neuron can have an enormous number of synapses. A spinal motor neuron covered by about 10,000 synaptic neurons from other neurons. You have 8,000 ending on its dendrites and 2,000 ending on its soma. So here on this slide, we're going to look at the different forms of the presynaptic neuron. For example, here's your presynaptic neuron, and here's your synapse. And of course, the signal is traveling from left to right, and hence your postsynaptic neuron that will receive that information. In B, you're going to have an axodendritic synapse, where you have the axon, the end of the axon, or the synaptic cleft, close to the dendrite, or synapsing with the dendrite. And your axon-somatic synapse, that would synapse with the soma. And of course, your axo-axonic, that will synapse with your axon. So those are the three visual aspects of your three forms of presynaptic neurons. Now when we look at the synaptic cleft, is a gap between neurons, and it was discovered by Raymond Yi Cajal through historical discoveries. But then back in 1931, no later than that, Otto Louis, he demonstrated that neurons communicated by releasing chemicals through this through the chemical synapse. Now he tested two frog hearts, and he exposed them, and he flooded them with saline. Now when he stimulated the vagal nerve of the first frog, the heart started to slow down. Then when he removed the saline from that first frog, he found that it slowed down the heart of the second one, and he termed it as vagus substance. Then he later renamed it acetylcholine, which was that very first known neurotransmitter. Now, electrical synapses do exist. We have some neurons, neuroglia, and cardiac and single unit smooth muscle. We do have these electrical synapses. They have gap junctions that join adjacent cells. The ions diffuse through the gap junctions from one cell to the next. Now, the advantage of having such a quick transmission is that there's no delay for release and binding of the neurotransmitter. And we have examples would be the cardiac and the smooth muscle and some neurons. But there's a disadvantage. The disadvantage is that they cannot integrate information and make decisions. The ability is reserved for chemical synapses in which neurons communicate by releasing neurotransmitters. Now, the structure of a chemical synapse includes the synaptic knob, which is a part of the presynaptic neuron that contains the synaptic vesicles. Many of the presynaptic neuron vesicles are docked at the membrane, while others far away constitute a reserved pool. The postsynaptic cleft, the postsynaptic neuron has no synaptic vesicles in the synapse and cannot release neurotransmitters. The membrane does not or cannot. The membrane does contain receptor proteins and ligand regulated ion gates. So here in this diagram, the structure of the chemical synapse. Here we have the axon. The top at the bottom here is our synaptic knob, which will be our presynaptic neuron, and down here we'll have our postsynaptic neuron. And this gap between the two is that synaptic cleft. Here are our vesicles containing the synaptic vesicles containing the neurotransmitters, and here are the receptors for these neurotransmitters, right on the membrane of the synaptic postsynaptic neuron. Now, more than 100 confirmed or suspected neurotransmitters have been identified. That's a lot. Now, neurotransmitters fall into 4 major categories according to composition. We have acetylcholine, amino acid, monoamines, and then we have neuropeptides. Now, with the acetylcholine, it's in a class by itself, and it's formed from acetic acid and choline. The amino acid and neurotransmitters, they include glycine, glutamate, aspartate, and GABA, which is our gamma-aminobutyric acid. Third one is our monoamines. They are synthesized from amino acids by removal of the carboxyl group. The major amino acids are epinephrine, norepinephrine, dopamine, histamine, and serotonin. The first 3 are in a subclass called catecholamines. There are epinephrine, norepinephrine, and our catecholamines. And our dopamines. Fourth group is our neuropeptides. Neuropeptides, they're chains of 2 to 40 amino acids, and they typically act in lower concentrations and have longer lasting effects than other neurotransmitters, and are stored in larger secretory granules. Now, they also have a function as hormones or as neuromodulators. Now, some are produced, but not only by neurons, but also by the digestive tract. Now, the neurotransmitters, they're synthesized by the presynaptic neuron. Then they're released in response to a particular stimulation. Then when they're released, they cross that synaptic cleft and they bind to the receptors on the postsynaptic cell. With that, they're going to alter the physiology of that particular cell. Now, a given neurotransmitter does not have the same effect everywhere in the body. Multiple receptor types exist for a particular neurotransmitter. So we have 14 receptor types for serotonin, which is a type of neurotransmitter. Now, a receptor governs the effect the neurotransmitter has on that particular cell. Neurotransmitters are diverse in their action. Some are excitatory, some inhibitory, and some the effect depends on what kind of receptor the postsynaptic cell has. Some hold ligandated regulated ion gates, and some act through second messenger systems. Now hopefully you understand what ligated means. When they say ligand-gated ion channels, they're referring to a group of transmembrane ion channel proteins which open to allow certain ions through. Okay. Alright, so neurotransmitters, they have diverse actions. The 3 examples of different modes of action are illustrated by the excitatory cholinergic synapse, an inhibitory GABA-ERGIC synapse, and an excitatory adrenergic synapse. Now the synaptic delay is the time from arrival of a signal at the axon terminal of a presynaptic cell to the beginning of an action potential in the postsynaptic cleft. So that's when that signal arrives just right at that terminal, and then right before, right at the beginning of when the neurons, the neurotransmitters have crossed that cleft and have binded to the receptors at the beginning there. That's considered that synaptic delay, and that's approximately 0.5 milliseconds for all the complex sequences of events to occur. Now the excitatory cholinergic synapse, it employs the acetylcholine as its neurotransmitter, and it follows a number of steps. When we look at the steps in figure 12.22, we see here that we have the arrival of a nerve signal at the synaptic knob. So here's our synaptic knob. So we have a particular signal that's going to arrive at the synaptic knob. And when that signal arrives there, it's going to open the voltage regulated calcium channel gates. Now the calcium enters the knob, and then it triggers the exocytosis of the synaptic vessels, releasing the acetylcholine. So of course we're going to have these so we have the calcium entering into the channel gates into the presynaptic ion, and we have these acetylcholine within these vesicles. So once the calcium enters in, it's going to trigger the release of them to the exocytosis stage, releasing them through the synaptic cleft. Now the empty vesicles are going to drop back into the cytoplasm to be refilled, and the synaptic vesicles in the reserve pool move to the active sites, and they're going to release the acetylcholine. Now acetylcholine, they're going to diffuse across the cleft and bind to the ligand regulated gates. which are going to open to allow sodium to enter the cell and potassium to leave. Now sodium spreads out along the inside of the membrane, producing a local potential called a postsynaptic potential. Now if it's strong and persistent enough, this triggers an action potential. Now, in inhibitory GABA-ERGIC synapse, it employs a gamma aminobutyric acid as its neurotransmitter. So the release of the GABA by the presynaptic neuron and its binding to the ion gates is a similar mechanism as for the cholinergic synapse, the synapse that we just previously talked about. It's very similar to it. Now the GABA receptor, however, is a chloride channel, and when it opens, the chloride enters the cell, as you see here. It enters the cell and it increases the negative membrane potential, then inhibiting any type of firing. So it's going to prevent anything from being signaled to the remaining parts of the neuron. And I have the description of what I was just talking about, the inhibitory GABA-ERGIC synapse. The third one is our excitatory adrenergic synapse. And that one is going to employ norepinephrine, which is one of our catecholamines, norepinephrine, as a neurotransmitter. Its receptor is not an ion gate, but a transmembrane protein associated with the G protein in a second membrane system. What is going to, the binding of the neurotransmitter, the binding of the norepinephrine to the receptor causes the G protein to dissociate from it. So here's our G protein, and here's our neurotransmitter. Once the norepinephrine binds to the receptor, it causes this G protein to be released. Now, once it's released, the G protein binds to the adenylate cyclase, which is an enzyme, inducing it to convert ATP to cyclic AMP. ATP, which we should know is adenosine triphosphate, and AMP we should know as adenosine monophosphate. Now, cyclic AMP can induce several alternative effects inside of the cell. One effect is to produce an internal chemical that binds to a ligand-regulated ion gate from inside the membrane, opening the gate. Another is to activate pre-existing cytoplasmic enzymes that affect metabolism. The last one is yet another is to induce genetic transcription so that new enzymes are produced. And here on the next slide, you will see a description of what occurs in that particular figure, in figure 12.23. Now, cessation of signals occurs when neurotransmitter stops being released, and when neurotransmitter already present in synaptic cleft is removed. Now, cessation of action potentials in the presynaptic nerve fiber stops the release of the neurotransmitter. Now, the removal of neurotransmitter from the synaptic cleft is occurred in three ways. We have diffusion, reuptake, and degradation. Now, with diffusion, the neurotransmitter leaves the synapse and enters the extracellular fluid. Now, in the central nervous system, the astrocytes, they absorb it and then return it to the neurons. With reuptake, the synaptic knob reabsorbs amino acids and monoamines by endocytosis and breaks them down with monoamine oxidase, which is an enzyme. Now the last one is degradation, and that's when the enzyme acetylcholine esterase, it breaks down the acetylcholine into acetate and choline, and then the choline is going to be reabsorbed by the synaptic knob. Now we have neuromodulators. Neuromodulators are hormones, neuropeptides, and other messengers that modify synaptic transmission. Now, they may stimulate neuron to install more receptors in the postsynaptic membrane, adjusting its sensitivity to the neurotransmitter. They may alter the rate of neurotransmitter synthesis, release, reuptake, and as well as the breakdown. Now we have enkephalins, and that is also in the neuromodulator family. Those are small peptides, and that inhibit spinal interneurons from transmitting pain signals to the brain. Then we have nitric oxide, which is a similar neuromodulator, and it's a gas that's released by the postsynaptic neurons in some areas of the brain. And here it stimulates the presynaptic neuron to release more neurotransmitter. Now one neuron's way of telling the other to give me more. And some chemical communication that goes backward across the synapse. Now the synaptic delays, it slows the transmission of these nerve signals. The more synapse in the neural pathway, the longer it takes for transmission to get from its origin to its destination. Now synapses are not due to the limitation of nerve fibers' length. We have gap junctions that allow some cells to communicate more rapidly than chemical synapse. Alright, so why do we even have synapse? It's to process information, to store it, and of course, to make decisions. Your chemical synapses are the decision-making devices of the nervous system. The more synapses a neuron has, the greater its information processing capabilities. The pyramidal cells in the cerebral cortex, they have about 40,000 synaptic contacts with other neurons. And the cerebral cortex has an estimated 100 trillion synapses. Now the ability of neurons to process information, store it, and recall it, and make decisions is called a neural integration. Neural integration is based on the postsynaptic potentials produced by neurotransmitters. Now, any voltage change that rises the memory potential closer to the threshold is termed excitatory postsynaptic potential. Any voltage change that hyperpolarizes the membrane and makes it more negative Then the resting memory potential is turned an inhibitory postsynaptic potential, IPSP. Now because of ion leakage, all neurons fire at a certain background rate even when not been stimulated. The EPSP, which is excitatory postsynaptic potential, and the IPSP, the inhibitory postsynaptic potential, only change the rate of firing. The glutamate and aspartate produce this EPSP, and the glycine and GABA, they produce the IPSP, our inhibitory postsynaptic potential. Now, acetylcholine and norepinephrine, those are excitatory for some cells, and they also inhibit. So they play both sides. Now here we have an example of the EPSP, and the IPSP. So hopefully you guys are going to review this particular diagram within your textbook. I believe it would be figure 12.24 that you will see in your textbook, where you see this is the first one where we have the voltage change that will rise, the member potential closer to the threshold. Here's your threshold. You have that voltage that will raise that member potential very close to that threshold. And this is what's called this EPSP, the excitatory postsynaptic potential. But our IPSP it will be our inhibitory. And that's going to be having a voltage which hyperpolarizes the membrane and making it more negative than the resting potential. So it's going to be going in the opposite direction. So you see it's getting more negative. Now one neuron can receive input from thousands of other neurons. Some incoming neurofibers may produce EPSPs, while others may produce IPSPs. Neurons' response depends on whether the net input is excitatory or if it's inhibitory. Now, summation is the process of adding up postsynaptic potentials and responding to their net effect, and it occurs in the trigger zone. Hopefully you remember where the trigger zone is. Trigger zone is right by it occurs in the axon hillox. Now the balance between the EPSPs and the IPSPs enables the nervous system to make decisions. We had the temporal summation, and that occurs when a single synapse regulates the EPSPs so quickly that each is generated before the previous one fades. And this will allow EPSPs to add up over time to a threshold voltage that would trigger this action potential. So we're going to have a lot of them adding up in order just for this action potential just to start. Now the spatial summation, that occurs when the EPSPs have several different synapses that will add up to the threshold at an axon hillox. For this, you're going to have several synapses that will emit enough sodium to reach this particular threshold that's needed. And the presynaptic neurons, they're going to cooperate to induce the postsynaptic neuron to fire. So here we have an example of the temporal and the spatial summation. Here we have the intense stimulation by one presynaptic neuron, and then triggering the EPSPs. They're going to spread from one synapse to the trigger zone, getting our presynaptic neuron to start firing. But in the spatial is when we're going to have stimulation by several different presynaptic clefts, presynaptic neurons, causing a spread from several synapses to the particular trigger zone. Then it's going to cause the postsynaptic neuron to fire. So in this diagram here we have stimuli. We have our EPEs that are being produced. We have our action potential, and we have our threshold, and as well as our resting membrane. So of course, at first it's at its resting membrane. When we get the stimuli, the several types of stimuli that are produced, is going to produce at least one action potential. One EPSPs. Now, if there's enough EPSPs that arrive at this trigger zone, faster than they start to fade away, then they can build up on each other and start to bring the neuron to a threshold and then trigger this action potential. Now, summation, facilitation, and inhibition are mechanisms that influence a neuron's integration of multiple inputs. Now, summation, which we talked about, is the process of adding up our EPE, our EPS, our ESPSs, and our ISPSs, and responding to their net effort, and it occurs in that trigger zone. Facilitation is a process in which one neuron enhances the effect of another. For example, when two neurons working cooperatively are able to induce firing. Now, the presynaptic inhibition is a mechanism by which one presynaptic neuron suppresses the action of another, often by blocking with an inhibitory neuroreceptor. So here we have our EPSPS, we have our neurotransmitters, but in here, there's no neurotransmitter that's being released. So at this point, it's blocking any type of inhibitory neuron from releasing any type of neurotransmitters. Now, the way in which the nervous system converts information to a meaningful pattern of action potential is called neural coding. The most important mechanism is the labeled line code. Each nerve fiber to the brain leads from a receptor that recognized a particular stimulus type, such as the optic nerve fibers. They're going to carry signals only from light receptors in the eye. Now, quantitative information, quantitative information about intensity of stimuli is encoded in two ways: in recruitment, another one is mechanism. Within recruitment, we have additional neurons are brought into play as a stimulus becomes stronger, enabling the nervous system to judge stimulus strength by which and by how many neurons are firing. So it's all going to depend on which ones are going to fire, and of course how many are firing. The second mechanism would be Another mechanism, it depends on the fact that the more strongly a neuron is stimulated, the more frequently it's going to fire, so that the nervous system can judge stimulus strength from the firing frequency of afferent neurons. So the absolute refractory period sets an upper limit to how often a neuron can fire. The theoretical limit of firing frequency is 2,000 action potentials per second, but the highest frequencies observed are 500 to 1,000 per second. Now here we look at the neurons' function and assemblies called neural pools, which may contain thousands to millions of interneurons. The function of the neural pool are partially determined by its neural circuit. Here they're going to control the rhythm of breathing, moving limbs rhythmically when walking. The information arrives at a neural pool through one or more input neurons. Now within the discard zone of an input neuron, that neuron can make the postsynaptic cells fire. In a broader facilitated zone, the neural synapse will still, with still other neurons in the pool, can stimulate them only with the assistance of other input neurons. A neural pool's neural circuit consists of the pathways among its neurons. A wide variety of neural functions result from the operation of four principal kinds of neural circuits. So we have diverging units, converging units, reverberating circuits, and parallel after discard circuits. So let's look at diverging circuits. Divergent circuits is one nerve fiber that branches in the synapses with several postsynaptic cells, so that input from one neuron may produce output through hundreds of neurons. The convergent circuit is input from many different nerve fibers, is funneled to one neuron or a neural pool. This type of circuit, it allows input from different sensory systems to be evaluated, such as correcting our balance. The reverberating circuits, neurons are stimulated in a linear fashion, but some have an axon collateral, leading back to the initial neuron and re-stimulates it. And we're looking at the diaphragm or also the intercostal muscles. The parallel after discharge circuits, within that particular circuit, you have an input neurons. They diverge to stimulate several neuron chains that eventually reconverge on a single output neuron. These chains different in total synaptic delay, meaning that their signals arrive on the input neuron at different times. Now there is no feedback loop, and once all the neurons have fired, output is ceased. Now the output neuron may continue to fire after the stimulus stops, as signals continue to arrive. So let's take a look at the four types of circuits. The divergent, we have one nerve fiber, and it starts to branch off from different several pulses synaptic cells, so that the input from one neuron may produce output through hundreds of neurons. Okay, so we're starting with one. And the convergent, what do we have here? we have input from different neurons. So here are neurons here. A lot of input from different neurons. I'm sorry. Then it's funneled down into just one type of neuron. This is our output. So we have input from several neurons, and it's funneled down into just one, where one neuron is giving output signals. Output signals to the particular target. Then we have the reverberating. We have neurons are stimulated in a linear fashion. So here's our neurons here, all getting stimulated in a linear fashion. But then some will have an axon that will correlate, leading back to the initial. So that some of them will actually branch off, leading back to the initial neuron to re-stimulate it. Then our last one is our parallel after discharge. This is where we have that input neuron here that will divert to stimulate several neurons to eventually reconverge on a single output of neuron. So we start off with one, it branches off and starts to stimulate other neurons, but eventually start to reroute and stimulate just this one neuron. So we get an output just from one particular neuron. Now the physical basis of memory is a pathway through the brain called a memory trace, or an engram. It is based on the ability of synapse to be added, taken away, or modified, which is called synaptic plasticity. Now during learning, synapses in a certain pathway become modified so that signals travel more easily across them, and this is called synaptic potentiation. So what kinds of memory do we have? We have immediate, short-term, and we have our long-term memory. Now with immediate memory, it's one of the three classes of memory, and it is the ability to hold something in mind for just a few seconds. Immediate memory is necessary for continuity of events and is especially important for reading. It might be based on reverberating circuits, which is the third circuit that we looked at. Now short-term memory, it lasts from a few seconds to a few hours. Now some people may experience this while they're studying for an exam or test, and they stick there just for a few hours until you get to the test and you actually forget. That's that short-term memory. Now the working memory is a form of short-term memory, and that allows us to hold an idea in mind long enough just to carry out the action, such as calling a telephone number we just looked up. It might even be just based on reverberating circuits. Now, somewhat long-lasting memories probably involve synaptic facilitation, which is induced by tetanic stimulation, where we have rapid arrival of the signals. Means that the calcium accumulates in this synaptic knob, then it causes the SPSPs to become stronger and stronger. Now memories lasting for a few hours may involve post-tetanic potentiation, and that's where we have the chemical levels. They stay elevated for such a long time, where it starts to signal that another signal is released, and it starts to burst out the neurotransmitter. So it will start to jog the memory of an event for several hours later. Our long-term memory lasts up to a lifetime and is less limited than the STM, which is our short-term memory, in the amount of information it can store. The two forms of long-term memory are declarative memory and procedural memory. Declarative memory is the retention of facts and events that can be put into words. Procedural memory is the retention of motor skills. Now, some long-term memory involves physical remodeling of synapses or formation of new ones through growth. Some long-term memory involves physical remodeling of synapses or formation of new ones through growth and branching of the axonal terminals and also the dendrites. Long-term memory can also involve long-term potentiation, in which NMDA receptors that bind the glutamate are simultaneously subject to titanic stimulation, and this will allow the entry of calcium as a second messenger into the capsid cells with three effects. Okay, the neuron produces more NMDA receptors, and it will make it more sensitive to glutamate in the future. It synthesizes proteins concerned with remodeling synthesis, and it also releases nitric oxide, which triggers more glutamate releases in the presynaptic neuron. So when we take a look at Alzheimer's disease, there might be some of us who know at least one or two people who may suffer from this particular disease, neurological disease. We know that at least 100,000 deaths occur per year from Alzheimer's disease, and that's approximately 11% of the population over 65 years of age. And by the age of 85, we have at least 47% that are suffering, that have died from Alzheimer's disease. Now we have memory loss for recent events, moody, combative, loss of ability to talk, walk, and as well as to eat. There's some side effects or some symptoms of Alzheimer's disease. This will show deficiencies in acetylcholine as well as in nerve growth. Now, when diagnosed, it's been confirmed at autopsy, and you have atrophy, which we know is like muscle loss of the various folds called the gyri in the cerebral cortex. And then we have formation of the beta amyloid protein from the breakdown product of these plasma membranes. The type of treatment that was done, they stopped the beta amyloid production, and that's due to very serious side effects that were occurring. So here we have someone with Alzheimer's disease. These folds, they're not supposed to be that deep, or wide, but with Alzheimer's disease, this is exactly what you're going to see. If you've seen a model of a brain, you will notice that there is definitely a difference with the sulci, which are the top parts of the folds of the brain, and then the grooves in between is called the gyri, and that's sunken in. It's very deep. It's not supposed to be that deep. Okay. Parkinson's disease, another neurological disease. It's a progressive loss of motor function, beginning obviously in the 50s and also in the 60s, and there is no recovery from them we know that who's some famous two people that I can't think off the top of my head. Michael J. Fox and the boxer. Oh, that's not good. I don't remember his name, but you guys may remember it. That has the Muhammad Ali. I'm sorry. Then we have degeneration. This caused from the degeneration of the dopamine releasing neurons. Now, dopamine normally prevents excessive activity in the motor centers. We have involuntary muscle contractions. That's where you have the shaking. Hands are constantly shaking, slurred speech, facial rigidity. Of course, their handwriting is not legible. The type of treatments that they use drugs and physical therapy. And they use, it's a dopamine precursor, which crosses the brain barriers. Now, bad side effects, unfortunately, affect the liver and the heart, just like most of the drugs that are out there nowadays. They are going to affect the liver and sometimes the kidneys. Now, surgical techniques, the to relieve any type of tremors. So that's the end of our chapter 12. If you have any questions, please go to the discussion board and post any questions, or if you see anybody that have posted questions and you would like to answer it, go ahead and do so at this time. Also, you need to be preparing for your quiz that will be on Sunday. This is a 10-point quiz, so just make sure you guys go over the points, go through the PowerPoint as well as the 15 minute video I posted on action potential, and start preparing for your quiz and as well as your test. Please do not procrastinate.